My name is Joan Griswold, and we are both with Genome Sciences Education Outreach at the University of Washington. And I'm a former teacher, middle school and high school science teacher. And we have been working on a Science Education Partnership Award grant for a number of years, and that's what is funding our work that you're going to see part of today. And Adam, will you introduce yourself?
I'm Adam. I use they, them pronouns, and I'm the Director of Education Outreach at Genome Sciences. I'm new to the project. Joan kind of got this going, and so it's been great working with her on this project. And on this project in particular, I work with the health teachers, but I'm really happy to be here with you all today.
What we are planning to do is I'm going to give you some of the background on the lesson at a teacher level, and then we'll walk through the lesson mostly as students. There are two simulations that are involved with the lesson, and so we'll do the first one all together, and then I'll just give you a break and let you do the second one totally on your own. We'll come back together and finish up with the end of the lesson, and then we'll go back to kind of teacher talk level at the end to debrief the lesson and think about some things about how you might use it and where and how other people have incorporated similar things. And you can share some of what you do with each other. As I mentioned, we have been working on two foundational units using type 2 diabetes as the phenomenon through which to teach gene and environment interactions. We have a set for health class, a set of lessons for biology class. They share the same enduring understandings, and they're very much complementary units. What we're showing you during the month of March mostly is what we call our expansion pack. And the expansion pack are a new set of lessons that build off of these foundational units in a slightly different way. So tonight is the microbiome. We've got sugar taste and type 2 diabetes, the ethics of access, and then brain snacks, which is goes on from there. So why type 2 diabetes? Well, we chose it because it's a very complex condition, and it leads to a lot of different question asking in a lot of different realms. So there are so many places that you can take this. There are no cut and dried answers. There are no cut and dried solutions, but it's a nice way to propose something for students, a real life problem in the world that needs solutions and needs solving and fixing, and having people think about all of the different ways that we can work together to figure this out. And then why the microbiome? Well, it turns out that there are some overlapping things, some concepts just around diet, the environment, choice, and then fiber is something that comes up a lot in both units, the importance of fiber in the diet. And plus, it's an opportunity for data analysis. As you'll see, we're hitting a new NGSS performance expectation about ecosystems, which is new for us. And teachers ask for it. So during our curriculum design sessions, teachers would, that was a topic that kept coming up as being of interest. So the driving question for this unit is how do food choices and the environment affect the gut microbiome in humans? And the learning objectives are that the gut microbiome is a diverse, very complex ecosystem, and the foods that we eat affect our microbiomes, and our microbiomes thrive on fiber. And diet can affect the microbiome in ways that can increase or decrease the likelihood of type 2 diabetes and other metabolic diseases. As far as NGSS goes, as I mentioned, this performance expectation is new for us, and it really gets into the changing conditions may result in a new ecosystem. In fact, thinking about this PE, I'm wondering how many complex ecosystems remain pretty stable, because stability in an ecosystem has to do, there are always things changing in any system, things in flux. And in this case, we're going to see how changes in this environment can actually change the populations in that environment. Students are looking at systems and system models. They actually perturb the system and see what happens. They're looking at stability and change. And they quantify this under their 2 conditions during the 2 simulations. And they analyze and interpret data. And then to hit the exact PE, we talk about evaluating, engaging an argument from evidence, where they're thinking about how fiber is what are the benefits of fiber and what's the evidence that supports that. So there are a few different ways of getting this lesson, and I don't want to confuse you. We're in this position where we keep creating new and different ways of doing things because teachers are in so many new and different ways of teaching things. And so what we've ended up with is we kind of have this collection of ways that you could teach this lesson. So what works well for one person may not work well for another. So what I've asked for you to have on hand or close by for this today is the student roadmap in the form, it could be either a Google Doc or a Word document. If you use this with your students, then you would need to make a copy of this document. And as students go through the lesson, they would be writing on this document itself. The student questions are part of this document. The data table is already incorporated in this document. So in a lot of ways, the student roadmap document is sort of a one-stop shopping. You've got everything there. It's also been made into a very pretty website, if I do say so. And the website is very, the websites are nice, they're slick, they are very good for students at home. Just clicking through, the students feel very comfortable with that. It is a Google site, so if you don't use Google in your school, then I hear that some schools don't even have access to Google sites. And as students go through this, they then would need to download a data table that they can be working with. And then they are asked questions on a student worksheet, which is in the form of a Google form. So there are a few more pieces that go along with this, but they both teach the same concepts pretty much in the same way, but just in a little different format. So this is straight out of the worksheet, the student roadmap, and I have done a lot of screenshots from that roadmap. So if you're at home and you can follow along, if you are lucky enough to have a dual screen or 3 screens, or I've seen these amazing configurations of many, many screens, which I now want. So you can follow along and see what we're doing. So this lesson has 7 parts if you're using the roadmap, and we will work through those. Before I get started, I do want to say that there's kind of a caveat, and this comes up a couple of times, is that when we perturb the microbiome, and just even talking about the microbiome, this is a new enough field that there's still a lot of questions, a lot of unsettled science out there. People are looking at it, but there are not a lot of answers. And one of the biggest questions is this question about causation correlation. So do certain diseases change the microbiome, or does a change in the microbiome play a role in causing that disease? And so when we're thinking about type 2 diabetes, that comes into play, but I just want to be upfront in saying that that is one of the biggest questions and sort of thoughtful pieces in this whole topic. And in this slide, there's a Google Docs, as I mentioned before. If you have a very sciencey question that you would like us to figure out the answer to, or ask some of our experts, we'd be happy to do it. And there's a link to that. And hopefully Adam can put that in the chat so everybody can have access to that Google Doc. All right, we are going to get started. And I imagine this as sort of a guided imagery. So if you bear with me, this is what I would like for you to do. For real, you're at home, if you feel comfortable, is to just shut your eyes, take a deep breath, and I'm going to read you something. So here we go. Imagine you are in a rainforest. It's hot, it's humid, and you can taste the air. There is green all around you and so many kinds of trees and plants you cannot count them all. Some have red fruits and some have tiny purple flowers. And the animals, there are birds, there are bats, squirrels, and monkeys. Mosquitoes are buzzing in your ear, and a giant Goliath beetle is quietly walking over some decaying leaves on the ground. The sheer diversity is overwhelming, and it is almost impossible to describe how all these living things are connected. Now imagine that same rainforest, but inside of your gut. It's still hot and humid, but instead of plants and animals and insects, you have microbes. These are tiny creatures that all look the same under a microscope, but are just as different from each other as birds and bats and squirrels and monkeys. It is a diverse, interconnected, and relatively unexplored place. Your gut is one of the most complex ecosystems on the planet. All right, you can open your eyes now if you actually have them closed. So students are introduced to the microbiome by picturing themselves in a rainforest. There's a little bit more to that reading, but that's where I left it for now. But through this, students are introduced to the idea that the microbiome is a very intricate web where nutrients cycle from organism to organism. One organism's trash is another's treasure, and they create a very complex web. It's a complex ecosystem. There are diverse populations there. All microbes are not the same. And we have a mutually beneficial relationship with the microbiome. Microbes have been on the planet way longer than we have. And so as we have evolved, we have work together with the microbes, and we mostly work, it works as a very beneficial relationship. And I will just show you, if you were using the online, if you were using the website, then students go to this sheet and they would fill out questions. So they would input their email address so that when they're finished with this form, it would get emailed to them. So you as a teacher could potentially have them share with you the email to show that they finished and show their answers. Or if you are going this route, you can always let me know. I can collect all of their answers and tell you who has filled out the form, where and when. So there's this, there are just a few questions about the microbiome or about the rainforest, and then it takes them to the next thing that we are all going to do, and that's watch a video, and then they will list a few things there. So we're not going to go back and forth to this student sheet. I just want to let you know that it is available, and that's how it works. Okay, so next up is a video that really talks about how the food you eat affects your gut, and we are going to watch it. All right, so after students learn something about the microbiome, or exactly that, about the microbiome, they participate in 2 different simulations. And for the first one, we are going to do your microbiome and random chance, where we are going to randomly get a starting microbiome, and then we are going to randomly perturb that microbiome in a few different ways. And I could use some help with this part if one of you has your data table accessible, because to be able to write down the data as we collect it, because we're going to do this all as a group. So for this activity, we need the microbiome spinner and the data table. So if you have the doc in front of you, you should have a data table. And let's spin this. Okay, which microbiome of the 4 are we going to get? All righty, we got microbiome number 1. So let's see. So let's take a look at microbiome number 1. On our data table, we are going to record the starting microbes in our microbiome by color. So I believe the blue is the first set. And if we look at microbiome number 1 right here, we've got 3 blue bugs. So if somebody could write a 3 in that column, that would be great. The next is green, I believe, and we have, so let's count them, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 green bugs. And orange, we have 3 different orange bugs or in your microbiome, bacteria. And then lastly is the gray, and it looks like we have a bunch of those. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 microbes for that. So, so if I remember correctly, which I mean, I think we had 3 blue. How many green did we have?
15, I believe.
Yeah, 15.
Thank you. Okay, and there were 3 orange?
3 orange.
Yes,
Okay, and
15
green.
gray.
15 gray. Okay, awesome. So that's great. We now need to disrupt our microbiome and see what happens. So I'm going to hit the microbiome disruption spinner and get that up. All right. So here's our random wheel. There are a number of things that can happen, and let's spin it. All right, we take antibiotics. So of our bugs that we have, that we already have on our data table, our microbiome, we're gonna lose 3 blue, we're gonna lose 4 green, we're gonna lose 3 orange, and 1 gray. So again, if I'm trusting somebody here to be recording this, and so spin 1, you can just do the math and put your new numbers of your microbiome there. Now we don't want to eliminate these because we might need another course of antibiotics, so we want to just resume and spin it again, and this will be our spin number 2. What's going to happen? Okay, we're starting a new medication, and that's going to affect our microbiome. We are going to gain 1 green and lose 2 orange. Now, if we've already, let's say, wiped out our orange, you can't go negative. You can't have, you know, a -3 number of orange. So then you're just at 0 because you can't have less than 0. All right, that was spin 2. Let's check out spin number 3. Uh-oh. We got food poisoning. We're going to lose 2 blue, 2 green, and then gain 1 orange. All right, and our last one. Diet change, bacon and peanuts for days. So in this case, we're going to lose 3 blue, we're going to gain 1 green and gain 2 gray. So that is our the last of our spins. Let's head back to the to here, which hopefully somebody has one filled out. And what we need to do is then look at the last amount. Actually, if somebody could just tell me, if I can go back, and I will write in, how many blue did we have at the end of that?
0.
Lost those, like in spin 1, maybe. How about green?
11.
Orange?
1.
And gray?
16.
Okay. So the next thing that we're going to do is an assay for butyrate. We heard a little bit about butyrate in the last video, and we'll learn more about butyrate in the next video. But our experimental assay is going to, we're going to need these numbers. So we've got 3 blue, and we get a +2 for every blue. Whoop. So that puts us, so that would be 6 blue that we have gotten at the end of that. How many green did we have again? 11.
Mm-hmm.
So that would be a -11 green. We get a +3 for each orange, and we had
1.
Orange. And then it turns out that the gray gray bacteria have no impact on our butyrate score at all. So our total butyrate score would be 6 plus 3 is 9 minus 11, which would be a -2. Again, you can't really have a negative butyrate score, so we're just going to call that a 0. So in other words, this microbiome didn't do anything for us, and the perturbations that happened to it didn't produce any butyrate for us, and we were kind of hoping for butyrate. So it turns out that the blue and the orange microbes are butyrate producers, orange a little bit more than blue. The green microbes actually inhibit production of butyrate. So that's why we are subtracting for each one of those. And then the gray microbes have no impact on butyrate production at all. So this is the first simulation, all random chance using the spinner. And then we are going to watch another shorter video. I think this is 4 minutes long, and it's out of Australia. All right, so now we know a little bit more about butyrate and resistant starch, which is a type of fiber. So we're still thinking about fiber, and it's a kind of fiber that is not easily digestible into glucose. So it resists digestion, but it's found in all sorts of plants and legumes and things that they talked about in the video. So now for simulation number 2, I'm just gonna let you go on your own for 5 minutes and look at what you need to look at. So I'm hoping that you have the student roadmap in front of you and something to write down on as far as the data table goes. And then for the second simulation, what you're doing is choosing a microbiome. So if you got to choose one of these 4 microbiomes, which would you choose? And then there are 9 different cards that you could choose from. 2 of them are your choice. So if you want to do something special and you can figure out how it affects your microbiome, you can do that. So again, start with the microbiome, figure out the number of bugs of each color in that microbiome. And then instead of spinning 4 times, you're going to take 4 choice cards. You can use the same one over and over if you want. You can mix it up. You can just experiment with the different cards there or make your own. And then we will meet back to talk about what you did, what happened when you got to choose your microbiome. Let's think about the simulations and how that went and what you chose.
I chose the 4th microbiome 4.
Okay.
I chose that one because it had a lot of the blue microorganisms and a lot of the orange microorganisms. I also chose to change by becoming vegetarian, then just going crazy with kombucha 2 different times. And then, you know, downing a lot of fiber. So
Okay.
I ended up with 86, a score of 86 butyrate.
Wow. Okay, I didn't know that one could get to 86 on that. So that's, you start with a great microbiome, you just feed it the right food, and that's a great butyrate score. So the students are asked these questions like, why did you choose the one you chose? What was the difference? How did the by random chance change when you got to make choices on your own? How are your butyrate totals different and what causes the difference? And then what dietary and environmental choices affect the gut microbiome? So those are some of those questions that are on the student sheet. And this again reinforces that performance expectation that organisms in stable conditions, organisms in changing conditions may result in a new ecosystem. So it's still complex. There's still, it's still a network where they're feeding off of each other, but it can be changed given a difference in inputs and the things that is sustaining that ecosystem. So the next, towards the end now that the students have done their simulations, is a data drop, or a place where students look at some data from a research study. And you could do this in a lot of different ways. I really like the Socratic seminar type system where you don't say a whole lot and just let students look at the graphs and figure out what they are, and maybe talk to each other and sort of work as a group to construct an explanation for what is going on here. So first, and in sort of asking a series of questions about this, the first level question is just like, well, what do these graphs show? You know, what's the title? What's on the x-axis? What's on the y-axis? Making sure that students kind of understand the structure of what they're looking at. And then the next step would be more of a evaluative question of, well, so what does it mean? You know, what are the data actually telling us about this? But here we have a study where the top 5 frequently produced metabolites were compared for healthy controls and people with type 2 diabetes. And what they're looking at here are the metabolites, or the end products of metabolism for the microbes in that microbiome. And for healthy controls, you can see that there's a lot of acetate and carbon dioxide. Those are the same 2 things, but healthy controls are producing a lot of lactate, propionate, and butyrate. So we've heard a lot about butyrate, but propionate and lactate are also thought to be beneficial end products, metabolites produced by different microbes. So people with type 2 diabetes are also producing acetate and carbon dioxide, but they also have ammonia, which is being produced, and hydrogen sulfate and ethanol are the next top 3. So it's a very much of a different compilation of the metabolites in their guts. The other thing that's interesting to notice on this is look at the y-axis and notice that the scale is very different. And when I was looking at it earlier, I was confused because I thought it should add up to 1, like that the total should be a total of 1, meaning that that's all of the metabolites in that system. But what this is they're making almost 80% of the microbes are making acetate. Over 50% of the microbes are making carbon dioxide. And then if we go to butyrate, about 42% of the microbes are producing butyrate. And then if you look on the people with diabetes, there's just a much smaller fraction of those metabolites being produced in the first place. Our graph tops off at 4/10 instead of at 1. The other semi-interesting thing that I am just going to throw out there about this study is that these healthy controls in people with type 2 diabetes are all male. So we could go into a whole nother lesson there. And they're all middle-aged and they're all of normal weight. So this is, they did control for a number of things, but again, another study that are looking only at the male gender, and then we are supposed to extrapolate to everybody else, which was there is a call within the biomedical community of diversifying the research subjects that participate in studies like this. And of the people with type 2 diabetes, they could not be on a diabetes medication such as metformin because that also impacts the microbiome. And as you see, they're also controlled for weight. So they were all of normal weight. So you could spend as much time or as little having students parse this graph and just seeing what is different between these 2 groups of people. And then after this, we go to another piece of data from the same study, which is this amazing graphic of a network, a microbiome network. And the reason I like this is because it really gets to the complexity. It's a little hard to see on this slide, but on the roadmap, you can go directly to the graph and you can kind of zoom in on your website and you can look at different things. But it shows the amazing complexity and the number of microbes that are involved in just this particular network. So in this study and on this network, the people with type 2 diabetes the microbes are designated with yellow, and then healthy controls are designated with blue. So you can see that they have just different populations and numbers of microbes in their guts. And so students are just asked to look at this. They're asked to find butyrate, which is in blue. So if you look carefully, you can see some butyrate producers down here. Here's butyrate one place, here's butyrate in another place. And students are asked to talk about the microbes that produce it. And so in this case, this is Roseburia, looks like produces butyrate. And this one is E. erectale. Wonder where one finds that one. Is also a butyrate producer. And we used in our little simulation 4 different microbes, and this one there are about 100 here, so there's a lot going on in this. Also it's nice to see that you have starch right here, that if you have, well, this Roseburia is producing butyrate, it is being fed by starch coming in here, feeding this microbe, which then acts on this one, which then produces butyrate. So you don't have to get very complicated with this, but it's just a way of showing the vast complexity of the system. So that's it for the data analysis portion of this lesson. And then lastly, I'm excited to share with you, this is brand new. So Cecilia Noecker, who worked in genome sciences for a while, she came and spoke with our curriculum design team a couple of years ago. And Adam has just recently interviewed her for this lesson. This lesson sort of grew out of her work and her presentation to the teachers. And so I just want to show you parts of it. It's about 15 minutes long, and we won't watch the whole thing, but it's been put into chapters as well, so that you can go to certain places and hear about Cecilia's thoughts on different things.
I'm here today with Cecilia Noecker, postdoc at University of California, San Francisco. Thank you, Cecilia, for spending time with us today to talk about your scientific career as well as the microbiome.
Yeah, I'm really happy to be here.
We're going to dive right in. So how did you get into science, and what interested you in science?
I had kind of a winding path into science. I was not, someone who knew from a very young age I wanted to do science. I was interested in a lot of things. I liked most of my classes at school. I was involved in sports and music and art. I liked learning new things. I liked problem solving. And in high school, junior year of high school, my biology class, we did some independent research projects. And that was kind of the first time that I realized that sort of the process of research was very exciting to me, and sort of saw what that could be. And from there was interested in exploring more, particularly biology and learning about the natural world. In college, I studied biology and math and statistics, and kind of went on from there then to graduate school and my current position. So.
All right, and I'll...
So in just kind of simple terms, can you describe like what the microbiome is and what it does for the body and what it does for health?
Yeah, absolutely. So the human microbiome refers to sort of the ecosystem of microscopic organisms that live in and on humans. And there are microbes sort of all over your body, on your skin and in your mouth, but in particular, most of them are live inside your gut, where they perform a variety of functions that are really like crucial for our life and health. So they help digest our food. They can synthesize vitamins. They sort of send signals to our immune system to help communicate like what a good microbe versus a bad microbe looks like to the immune system. And they can help regulate our metabolism. And we're still sort of learning new ways that sort of signals sent by these microbes inside of us interact with our own host cell functioning.
I'm curious.
All right, and there's some interesting bits on brain health. And I'm looking for the career section because this is also.
What kind of careers can research into the microbiome lead to?
Yeah, I think this is a great question because I think that many different careers in the near future will be thinking about the microbiome in what they do on a regular basis. So obviously scientists are studying the microbiome, but doctors, the microbiome is increasingly being integrated into the practice of medicine. So when you're prescribed antibiotics, hopefully the doctor will more frequently in the future suggest that you should consider a probiotic or consider ways to sort of restore your microbiome after taking that antibiotic. But also things like nutritionists thinking about how our food shapes our microbiome, pharmacists. So we know people with different microbiomes can metabolize drugs in different ways. So probably in the future, you might get sort of a microbiome test to determine what dose of a drug you should receive. And even things like engineering and design, we know that our microbiomes are shaped by the spaces we spend time in and the surfaces we encounter. So things like antimicrobial materials and clothing, I think in those careers, people will be thinking more about how those things interact with our microbiomes as well.
Well, I never would have thought clothing, but I guess it makes sense considering we have our microbiome is also on our skin as well.
Yeah, that's obviously right. More relevant to the microbes on your skin, but yeah, like they can talk to your immune system. So these things do end up kind of all connected.
That's the video with Dr. Noecker, and feel free to, we would love some feedback and just know how you might use that with your students. And again, we were looking for a career tie so students can see who might do this work and what other possibilities are there for people who are interested in this field. And then, so that's basically the end of the lesson. The takeaways for this lesson are that microbes in our gut break down food we cannot break down, and some of the byproducts of metabolism, like butyrate, are helpful to our health. A high fiber diet can promote the growth of a healthy microbiome. And again, it's hard to know about causation. Does disease change the microbiome, or does a change in the microbiome contribute to disease? So how do our diets and actions change our microbiomes, and how do changes in the microbiomes change our diets and actions? So it's this very circular thing, and there's a lot to learn around this as well. And we want to be careful not to come down on the causation side of things. And but we do know that our dietary choices influence our health, specifically around fiber. So that's the lesson. Right now we'll do breakout rooms, maybe 3. We can have 3 or 4 people in each one. And here's some topics to talk about. You don't have to plow through each one if there's one that appeals to more than others. And I'll put these in the chat, or between Adam and me, we can get them in the chat.
Joan, we had an interesting discussion, and we were going to ask you about something in it. No, I can't remember what we said. About
the reason why the starch resistant foods.
Yeah, like we've read about starch resistant foods, things that you can make starch resistant by first cooking it and then cooling it. What is that all about?
I wish I could tell you. I'll add that to our, we asked, we were working with a dietitian a couple of years ago when we went to interview him, and it was the first time I had heard about, actually he was a nutritional researcher, and it was the first time I had heard about starch resistant foods. And Right, like cooking a potato and then recooling it changes the molecular structure so that it's not digested in the same way, literally. I mean, so that your body can't just liberate the glucose molecules. So the bond structure that gets shifted, I don't know the specifics of. I can circle back to him and ask though, because it's a hot new topic and I'd like to know more about that too.
Well, and it was an interesting kind of discussion about if you know, that stickiness of talking about food when you have kids who live in poverty. And if you talked about, you know, what is available, what do you have at home, what do you eat? And I mean, you could talk about fried rice. And if you could take rice that was previously cooked and then cool it and then heat it again, wouldn't that make a resistant starch out of it? And kimchi fried rice, that would be like the perfect, you know, gut bomb. So.
Yeah, that's good. And I'm glad that you were talking about how just talking about food, let alone diet, it is requires a lot of sensitivity on all sorts of different levels. And so did other groups talk about that, or would anybody else like to share anything that they talked about in their group?
That came up in our group regarding kind of eating disorders and just the complicated nature of food in general. And I think that's one of the things Joan and I have been thinking a lot about with regards to all of our diabetes curriculum, is kind of making sure that we're not exacerbating the problem. And so we do have plans to run this curriculum by the Emily Project here in Seattle to try to get some kind of feedback and add tools to the curriculum to give y'all the tools you need to have those discussions in a way that empowers people with information, rather than like, we're just not going to talk about this because of, you know, it makes us uncomfortable. Like ultimately that doesn't help us solve the problem or like work our way through those issues. And I'm not sure if the rest of my breakout room made it back. I think we might have lost them, but yeah, that was one of the things that we really talked about in our group. Oh, they did make it back.
We
okay.
are here.
Okay, great.
And along those lines, I think interesting that we talk this, and this lesson in particular talks a lot about choice and less about structure. And we try to talk a lot about structure in the diabetes curriculum because we're talking about choice and making food choices that involve high fiber. But those choices have to be in your environment for you to even be able to make that choice. So if you live in a food desert, or if you are eating a lot of fast food, not because that's your favorite thing, but that's quick and easy and cheap, and, you know, going to the farmer's market just isn't in your wheelhouse then that's something that needs to be addressed too. So students don't feel like, oh, I'm just making lousy choices, but it's like, what do you even have offered? And a good place to think about that could even be the school cafeteria, if we're ever back in schools with cafeterias. You know, what sort of choices do you have there? And are those the choices that you would want to feed your microbiome? Lenore, can I put you on the spot? Because you've just taught this with your class, right? And so I'm not sure if you were able to share, or do you have any words of wisdom for the rest of the group?
I did tell my group I ended up splitting the lesson in half and having it over 2 days. So they went up through the random part the first day, and then they did the choices and the data drop on the second day. They enjoyed and understood the videos, but they did have trouble with the simulations, and some said they didn't know how to use the spinners. So
okay.
Yeah. And others, calculating the butyrate wasn't clear for them. They just took the total population and then -2 or +1. I went, no, that's not what the instructions said. But overall, I think they got the main idea. But yeah, they did need more than one day.
Yes, this would be a lot, even for 2 days. And I know things get really stretched out online. Great feedback. Do any of you teach the microbiome currently? Or use more traditional methods to get to ecosystems, which is a more traditional topic?
I touch on it, but in my human biology class, when we're talking about the digestive system, we talk about a little bit about the gut biome, but had never thought about teaching it as just an ecosystem or ecosystem related thing.
And I did ask my students, what did they think about this as an ecosystem, since it's not something we normally would teach about. And they were, yeah, this is something we never thought about. And for Health Academy students, yes, we should learn about what's inside of us. So very receptive.
I would love to extend this with some information about how different bacteria interact with each other in the gut, as some of those interactions. We focus on that a lot when we're looking at sort of large scale ecosystems. And I think that would be really cool, just how, the learning a little more about how our good bacteria kind of keeps the bad bacteria at bay, and learning some of those mechanisms it's introduced at the beginning, which is great. And I would love to see, to kind of spin off that.
Yeah, that would be a really interesting extension. I like that.
And I think that's one of those cutting edge questions that people have about how all these different microbes interact with each other. And so in my interview with Cecilia, she was talking about her favorite gut microbiome or microbe is this Egerthella lenta, which just culturing it is really, really difficult because it requires blood, and it requires like very specific culture conditions. And so you can imagine like growing bacteria in your colon is very different than growing it in a test tube. And so oftentimes like mimicking the gut within an experimental situation is extremely difficult. And because you have skin that has bacteria all over it, you're constantly contaminating everything. And so there's a great question in the question wall about C-sections versus vaginal birth. And like, it's just, you're going to be colonized by microbes. It's just a matter of which ones are you getting colonized by. And I think within the lesson, having that random element, like that's random, right? We don't choose whether we're vaginally birthed or C-section birth. And so right off the bat, we're not necessarily able to make choices for what microbiome we're going to get. And that's happening constantly as well. I mean, I have been making kombucha as well. I think it got infected with something, and that did not help my microbiome. And so you can see there's just so many working pieces there, and we don't know as much as we do know, or way more than we do know.
And when we're just up against the end, when you were talking about the video with Cecilia, I thought you were going to mention the gut-brain interaction, because there's a really super interesting connection between our feelings and our emotional states and the gut bacteria that we have. And so that's another really interesting place to go in the future. We are at the end of our time, and I want to honor your time. I also put in the chat a link to a folder, and the folder has everything that you had access to today, including the PowerPoint that I used. So if you want to use that, you can. I think you will probably need to make copies of all of those things. You don't have editing privileges, but I believe you could copy anything in that folder. And if you don't have access to Google, let me know, and I can send you a PowerPoint version. So thank you all for attending our first Wednesday session, and hope to see you in the coming weeks, and for very different sorts of offerings. Really appreciate your time and attention this afternoon at the end of a workday.